Hello PG students. Let's begin with the third subunit of bacterial systematics, that is three-domain classification system. The Earth is 4.6 billion years old, or maybe more than that, and microbial life is thought to have first appeared in between 3.8 and 3.9 billion years ago. In fact, 80% of Earth's history was exclusively microbial life. Microbial life is still the dominant life forms on Earth. It has been estimated that the total number of microbial cells on Earth on the order of 2.5 into 10 raised to 30 cells somewhere, making it the major fraction of biomass on the planet. Now let's see three-domain classification system. The three-domain system was first introduced by Carl Woese in 1990. So it is also called as Carl Woese classification. The three-domain system proposed by Woese and others is an evolutionary model of phylogeny. Phylogeny means which refers to the evolutionary relationships between organisms. It's an evolutionary model of phylogeny based on differences in the sequences of nucleotides in the cell's ribosomal RNAs, as well as the cell's membrane lipid structure and its sensitivity to antibiotics. Okay. Now, why comparing rRNA structure is useful? It's because rRNA molecules throughout nature carry out the same function. Their structure changes very little over time. Their similarities and dissimilarities in rRNA nucleotide sequences are a good indication of how related or unrelated different cells and organisms are. One more thing. The main difference from earlier classification system is the splitting of archaea from bacteria. And so, the three domains of Carl Woese classification system include archaea, bacteria, eukaryote, and kingdoms. Now, Carl Woese, he used 16S ribosomal RNA as a chronometer. Why? Because it is universally distributed, means its presence in all species, or it is present in all species. It is functionally similar in all organisms. It can change its sequence slowly. Its sequence can be aligned or matched up between two organisms. Now let's see that there are various hypotheses as to the origin of prokaryotic and eukaryotic cells. Because all cells are similar in nature, it is generally thought that all cells came from a common ancestor cell termed as last universal common ancestor, LUCA, L-U-C-A. These LUCAs eventually evolved into three different cell types, each representing a domain. More recently, various fusion hypotheses have begun to dominate the literature. One proposed that the diploid or 2N nature of the eukaryotic genome occurred after the fusion of two haploid or 1N prokaryotic cell. Other proposed that the domains Archaea and Eukarya emerged from a common archaeal eukaryotic ancestor that itself emerged from a member of the domain bacteria. Also, some of the evidence behind this hypothesis is based on a superphylum of bacteria called PVC. They are the members of which share some characteristic with both archaea and eukaryotes. There is a growing evidence that eukaryotes may have originated within a subset of archaea. Now, in an event, it is accepted today that there are three distinct domains of organisms in nature. There are bacteria, archaea, and eukarya. So as you can see, 3 domains: domain Archaea, domain bacteria, domain Eukarya. See, this is the tree diagram. You can see. Let's see one by one. Domain Archaea. The Archaea domain includes all prokaryotic cells. They lack nuclear membrane and have distinct biochemistry. They contain RNA markers from bacterial cells. Now they are also considered as the oldest species of organisms on Earth. They can survive in extreme harsh environments and include methanogens, extreme halophiles, and hyperthermophiles that differentiated them from other domains. Now, one reason why this is because is that the ether containing linkages in the Archaea membranes is more stable than the ester containing linkages in the bacteria and Eukarya, and are better able to withstand higher temperatures and stronger acid concentrations. Understood? Now, the cell wall of Archaea lacks peptidoglycan. Unlike the bacteria and Eukarya, the Archaea have membranes composed of branched hydrocarbon chains, like many also containing rings within the hydrocarbon chains, attached to glycerol by ether linkages. I'll show you the figure. Archaea are not sensitive to some antibiotics that affect the bacteria, but are sensitive to some antibiotics that affect the Eukarya. Fine. See, this is the membrane lipids of Archaea, and here you can see the ether link, glycerol group and the ether link. Understood? This is the difference. Now Archaea, they have 3 phyla: Crane Archaeota, Euryarchaeota, Con Archaeota. Crane Archaeota, they can survive at extremely high temperatures and extremely low temperatures also. Euryarchaeota, some of them are known as extremely halophiles, which can inhibit highly saline environments. Con Archaeota, it includes all those species which were found in a single hot spring. The place is Obsidian Pool, present in Yellowstone National Park, USA. Now some more examples for archaeal organisms: methanogens, halophiles, thermoacidophiles. Methanogens, which produce the gas methane; halophiles, which live in very salty water; thermoacidophiles, which thrive in acidic high temperature water. Okay. Moving ahead, domain bacteria. These are also prokaryotic cells with bacterial rRNA and contain diacyl glycerol diester lipids in their membrane. They are also called as eubacteria or true bacteria. Their cell membrane contains ester linkage between unbranched fatty acids chains and glycerol. Their cell wall is made up of peptidoglycan, and bacteria are sensitive to traditional antibacterial antibiotics but are resistant to most antibiotics that affect Eukarya. Now there are 5 phyla of the bacterial domain: Proteobacteria, Cyanobacteria, Eubacteria, Chlamydias, Spirochetes. Let's see Proteobacteria. Example of Proteobacteria is E. coli, Salmonella typhus, Legionella, Heliobacter pylori, which is causing many ulcers. Okay, there are many other things also. N gonorrhea, which causes gonorrhea. Cyanobacteria. Example of cyanobacteria is photosynthetic blue-green bacteria, which produces O2 gas. Eubacteria. Example is Clostridium. Tetanus botulism. Bacillus, Mycoplasma. Chlamydias. Example Giardia chlamydia, which causes STD. Spirochetes. Example spiral bacteria that causes syphilis or Lyme disease. Now one more example is actinobacteria. They are gram-positive bacteria including Bifidobacterium animalis, which is present in the human large intestine. Okay, next domain Eukarya. These are eukaryotic cells with a membrane-bound nucleus. Their membranes composed of unbranched fatty acids chains attached to glycerol by ester linkage. See again over here. In bacteria, it's the same. In eukarya, it's the same. They lack peptidoglycans on their cell wall. Eukarya are resistant to traditional antibacterial antibiotics but are sensitive to most antibiotics that affect eukaryotic cells. Now, there are 4 kingdoms of Eukarya: Protista, fungi, plantae, animalia. In the last lecture, we have seen these kingdoms. I hope you remember and you are working on the assignment. Okay. Protista, these are slime molds, euglenoids, algae, and protozoans. Fungi, these are sac fungi, club fungi, yeasts, and molds. Plantae, these are mosses, ferns, conifers, and flowering plants. Animalia, these are sponges, worms, insects, and vertebrates. Okay. See, this is the membrane lipids of bacteria and eukarya. Here you can see glycerol and the ester linkage. In Archaea, it was ether linkage. This is the basic difference. So I hope the concept is clear. Now you can understand the differences, the classification. Okay. Niches. Now niches with respect to biology means the way an organism fits into an ecological community or ecosystem. Let's see how. Now each of the 3 cell types tends to fit into recurring specialities or rules. First, bacteria, it tends to be the most prolific reproducer, at least in moderate environments. Next is archaeans. They tend to adapt quickly to extreme environments such as high temperature, high acids, high sulfur, etc. Now this includes adapting to use a wide variety of food sources also. Next is eukaryotes. They are the most flexible with regard to performing cooperative colonies such as in multicellular organisms, including humans. Okay. I hope the 3 domain classification system is clear to all. Start working on your assignment also. If you have any doubts, any questions, do ask me. Thank you.